So hello guys, our topic is instrumentation of UV. So in instrumentation, the essential components are source, lens, mirror, slit, which is monochromator, then the sample holder, detector, and recorder. So here is the diagram of single beam UV spectrometer and double beam UV spectrometer. The source of light used in UV is should be stable, intense, and have range of spectrum from 180 to 360 nanometer. It should be stable with no fluctuations and have sufficient intensity. High and uniform intensity is also required, and it should not be fatigued during use. And the source of light used in UV is hydrogen discharge lamp. In hydrogen discharge lamp, high pressure is used where hydrogen molecules, excited hydrogen molecules, are produced which emit the radiations. The hydrogen lamp cover the range of 3500 to 1200 Armstrong. They are stable, robust, and widely used. The second source is deuterium lamp, and the third is xenon discharge lamp. In deuterium lamp, deuterium is used in place of hydrogen, as in hydrogen discharge lamp. It is 3 to 5 times more intensity, and it is expensive. Xenon discharge lamp. In xenon discharge lamp, there is 10 to 30 atmospheric pressure, and two tungsten electrodes are used. It also has high intensity than hydrogen discharge lamp. Fourth one is our tungsten lamp. The tungsten lamp is similar to electric light bulb. Tungsten filament is used, which is heated, and the radiation is produced. It is also stable, robust, and easy to use. Mercury arc. Mercury arc contains mercury vapor and offers sharp bands. The spectrum is not continuous, hence it is not widely used. So now next part is monochromator. In monochromators, lens, mirrors, and slits are used. Lens are used to converge the light. Mirrors are used to focus, reflect, collimate light beams in spectrophotometers. Then slit is an important part of the monochromator, where the polychromatic radiation is converted to monochromatic radiation. To achieve this, entrance slit, exit slits are used.
Second instrumentation is monochromator. A monochromator is used to select the wavelength at which absorb measurement is to be made. It is a device used to isolate the radiation of desired wavelength of the continuous spectra. The former was original component, but now most modern instruments are employed gratings. There are three components. First is entrance slit, second is depressing element, third is exit slit. There are two main types of chromators, that is prism and diffraction gratings. When we see prism, when a beam of monochromatic light passes through a prism, it bent refracted. The amount of deviation is depend on wavelength. Blue light being refracted more than the red. Prisms are made of quartz for use of ultraviolet region. Since the glass absorbs wavelength shorter than about 330 nanometer, glass prisms are preferable visible region of spectrum as the diffraction is much greater than obtained quartz. Prism produce non-linear diffraction, with long wavelength being less efficient. Second type is grafting. The depressing element is the monochromator of most modern ultraviolet, visible, or infrared spectrophotometer. It consists of very large number of equispaced lines, about 200 to 2000 per nanometer, ruled on glass blank coated with film aluminum, produced by modern holographic techniques. They can be used as transmission gratings or when illuminated as reflection gratings. Rotation of grafting permits appropriate wavelength of spectrum to emerge from the exit slit of monochromator. First transmission grating and second is diffraction or reflection grating. Now we see the advantages of grating. Grating gives higher linear diffraction compared to prism monochromator. Can be used over wide wavelength ranges. Constructed with material aluminium resistant to atmospheric moisture. No loss of energy. High polished surface like aluminium. It can be used over considerable wavelength ranges. Grafting and strong, long lasting. Now we see sample holder. The sample holder to be analyzed is mostly in solution from it is held by sample holder, also known as cuvettes. These are made of quartz or fused silica, which can easily allow passage of desired radiation. Most common cell length of sample holder is 1 centimeter. It should fulfill main conditions. First is it must be uniform in construction and thickness must be constant. Material of construction should be inert to solvents. Commonly used cells are made of quartz or fused silica.
Fourth instrumentation is detectors. Detectors used in UV visible spectrophotometer can be called as a photometric detectors. These are those devices which convert light sources into electrical signal which finally displayed by recording system. Most commonly used detectors are photocell, photomultiplier tube, photovoltaic cells and barrier layer cells. Photocells. When the light is incident upon a photocell, the surface coating emit electrons. The electrons are attracted and collected by anode. The current which create between cathode and anode is regarded as a measure of radiation falling on the detectors. This photocell is more sensitive than photovoltaic cells. If quartz or fused silica windows are used, range of photocell can be increased through the near UV and into a far UV region. Photomultiplier tube. It is the most commonly used detector in UV spectrophotometer. It is a combination of photodiode and an electron multiplying amplifier. The tube consists of an evacuated tube which contains one photocathode and 916 electrodes known as diodes. The surface of each diode is of BeCu CuSb. Working. When a radiation falls on metal surface of photocathode, it emits electrons. The electrons are attracted towards a diode which is kept at a positive voltage. When electrons strike first diode, more electrons are emitted and are attracted towards second diode and so on. This process is repeated until a shower of electrons reached the collector. The number of electrons reaching the collector is the measure of intensity of light falling on the detector. Photomultiplier tube is extremely sensitive and fast in response. The transmit time is in the range of 10 to 100 micron per second. Photovoltaic cell. It consists of semiconductor which is in selenium deposited on strong metal base like iron. Then a very thin layer of silica or gold is sputtered over the surface of semiconductor to act as a second collector electrode. Working. Radiations falling on the surface produces electrons at the selenium silver surface. A barrier exists between selenium and iron which prevents the electron from flowing into iron. Thus electrons are accumulated on the silver surface. This produces an electrical voltage difference between silver surface and base of the cell. It is generally used for instruments like photometers, which allow a wide band of radiation to strike the detectors. It is a simple in design and does not require any external power supply. With time, photovoltaic cells become useless because of the transformation of selenium layer. Thank you.